I would now like to introduce the first presenter of this block, Dr. Danielle Beckman. Dr. Beckman is a neuroscientist at the University of California, Davis, who works on primate models of neurological diseases. Her research includes studies of the neurological consequences of viral infections, including Zika and SARS-CoV-2, and she has produced award-winning imaging of brain infections. Today, she will present on neuroinvasion and neuroinflammation of SARS-CoV-2 as mechanisms of brain fog and cognitive dysfunction in long COVID patients. Welcome, Danielle Beckman.
And thank you very much, everyone that stuck to the last session. I know it has been a very long and intense day, 2 last days, but I very much appreciated everyone staying. I just want to briefly say that I'm going to share I have a lot of text. I put a lot of information on my slides, and I'm going to share as soon as the meeting's over. I'm going to share with the organizer, and I'm also going to share online. So if you want to don't worry about taking notes or anything. Everything is going to be available. So just moving on to the science. Okay, hold on. Yeah, there it is. So just talk. It's really hard for me to start, like where to start when we talk about viral infections in the brain. But I think the major point I want to talk is that the brain is not only about neurons. We have the glial cells. We have this, like showing this image here, like not only neurons are important in the brain, we actually have this homeostasis of glial cells, so microglia, oligodendrocytes, and astrocytes. These cells are working in balance together with neurons. And importantly, all these cells can be target for viral infections in the brain. So we know that, as I showed here in this table on the right, like different types of viruses can infect different types of cells in the brain, and including DNA and RNA viruses as well. And like these different viral infections can induce, different viruses can induce different types of pathogen-mediated effects in the brain, and also cause different neurological effects. And this is all very new, very recent in terms of neuroscience. Like we are starting to look at this, how these viruses can impact the cells and can induce cognitive decline and other markers of neurological impairment. What I think is also important, and I want to mention this study from last year, which 400,000 patients, is that we are learning more nowadays that virus can actually actively infect the brain, and this can have a big impact in the development of neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease. And we know that even a single viral infection can make a person more vulnerable to a neurodegenerative disease up to 15 years later. So it's really hard to trace back, like how can we correlate this, and we are learning with that. But we know nowadays that vaccination are able to improve these outcomes. So vaccinations can induce the presence of neurodegenerative disease associated with viral infection. And we also know, it's very interesting, it's very recent, but we know some antiviral therapies can actually provide improvement of degeneration and can even slow down neurodegenerative diseases. And one of them, and I think is interesting, is the Paxlovid, which is a drug that has been using for COVID. We know that is a drug that actually crosses the blood-brain barrier and can enter the brain. So it can offer a potential goal in the future, something we can explore, like to reduce and finish viral infection in the brain. So that might be interesting for avoiding neurodegenerative diseases in the future as well. Okay, so all these viruses can infect the brain, but why SARS-CoV? I keep talking online. Why SARS-CoV? Why coronaviruses in general are so dangerous for the brain? And there's a couple of reasons for that. I like to mention that there are several coronaviruses that are characterized, that we know they're around, but only 7 of them are known to infect the humans. So just the 7 that are shown here in the table on the left. And what I think is interesting that all 7 coronaviruses that are known to infect humans are capable of infecting the brain as well. So coronaviruses are very neurotropic. And I don't think it's a coincidence that all the recent pandemics we have in the last decades were all caused by coronaviruses as well. So neurotropic viruses are definitely causing more damage than the most of the virus circulating around. And coronaviruses are especially dangerous for the brain because they are able to access the brain through multiple different routes. So I think a lot of people are familiar with the idea that olfactory impairment caused by, which is like a letter A here in this image, and viral infection through the peripheral nerves and olfactory bulb, a lot of people don't realize that the loss of smell is actually nerve damage. And it's like you're losing these neurons, and they are like directly connect these nerves in the olfactory bulb by directly connect to the neurons in your brain. And it's very easy and very fast for them to track and just enter the brain through this route. But that's not the only route that coronaviruses use to enter the brain. They also can, like shown in B, C, and D, they can damage the cells in the blood vessels. Immune cells can hijack and just cross and directly induce their inflammatory responses, or you can even have like D, viral opening of tight junctions. So very few viruses are coupled to do all these types of different approaches to directly infect the brain. So this is for me why one of the most important reasons why coronaviruses are really neurotropic. And I also like to mention that this study, for example, from 2006, so even before SARS-CoV-1 or SARS-CoV-2 being appeared in our lives, we see that actually we know more or less through animal studies, we know how the disease usually progresses in the brain. So we know that in acute phase, we have viral replication, active viral replication happening in the brain. We have viral antigen expression as well. But the clearance of the clearance of the infection is the challenging part, and we know is often characterized by extensive demyelination as well. And neurons are the preferent type of cell for coronaviruses to enter the brain and to spread through the brains. And actually, neurons are excellent for the viruses because they are highly complex and they have this morphology of the dendrites and axons that allow the virus to propagate fast between different neurons, then like spreading through synapses, and can really go really fast to multiple regions of the brain. So I also think it's important to mention that coronaviruses, and this again is all characterized from 2006. So we know that even before SARS-CoV, MERS arrived, but we know it's a big challenge to clear coronaviruses from the brain because they are RNA viruses and they induce a lot of immunological responses in the brain. So you can see infection here in the first, and the control clearance will be like a phase of the acute phase. And then challenge is the long-term control. We know that patients with long COVID, they have CD4 cells and B cells impairment in the brain, for example. So the long-term control is the big challenge. Like even if you don't have new virus being generated in the brain, it's a big challenge to clear this virus being produced in the already infected neurons. So this is a challenge, how to clear that. And there's a lot of indication that we actually have viral persistence in the brain of the long COVID patients. And going to the microscopy, which is actually what I work with, we have been characterizing how neurons are the major target for SARS-CoV infection in the brain in our monkey models. So I work at the Primate Center in California. And in infected animals, we see clearly, as you can see in the image in the right here, we see clearly active viral infection, so viral replication happening inside the cell body of the neurons. And we don't see that in the control animals. And when we characterize this, how this infection is happening, we see that neurons, and here in the right, you see a graph for quantification of the pathology. So you see NEWN is a marker for neurons, GFAP for astrocytes, and IBA1 for microglia. We see all these three cell types can be affected by SARS-CoV, but for some reasons, like I explained before, neurons is really more like the favorable route for virus because it's so much faster to propagate, and it's really hard to clear the virus from the neurons. Just showing briefly again, like you can see here the virus, so double-strand RNA in red in the left, and you can see how the presence of the virus really reduce their neuronal protein. You see like these neurons are like close to dying or dead already. And you can see in the video in the right how you can actually see spike protein being very present in the dendrites and axons. And these animals are all non-vaccinated, by the way. And you see active viral infection happening exactly in the cell body. So the virus really uses the dendrites and axons as an entry route. And we see active viral replication in the cell body. And later on, the virus uses the dendrites and axons to propagate from a neuron to another. So characterizing this, as this image shows exactly the same thing. We see double-strand RNA, which is active viral infection in the cell body of neurons, and we see spike protein spreading mostly everywhere in the neuronal body. And we can quantify this type of damage using 3D reconstruction morphology. And you can see in this image like how you compare a control healthy cell body versus a control of the neuron with double-strand RNA from the SARS-CoV virus being replicating inside the virus. In the dendrites, we can calculate the spike protein, and you can see this loss of volume occurring in the control versus infected, control animals versus infected monkeys. I also like to point that this, as I said, like olfactory roof is very convenient for the virus to replicate because it accesses the brain so fast. And a lot of people don't realize that a lot of regions such as the entorhinal cortex that I've said before, or the orbitofrontal cortex here in green, are in the frontal cortex are primary and secondary olfactory paths. So these are brain regions that are controlling memory, cognition, not only controlling smell and olfaction. These are actually much more complex regions, and they are highly infected. And we see an increased infection in aging animals, in our aging monkey, compared to the young monkeys as well. I want to briefly mention this study because I think it's really important. It's a study from 2022 that they compare patients before and after. So they have MRI from patients that before they get SARS-CoV infection, and they compare with controls before and after as well, and in an interval of 3.2 years. And I think what the major findings that they found in this study is exactly atrophy, brain atrophy, and degeneration in these patients in the same regions that we are also observing microscopic changes and active viral infection as well. So in this study, they really said like one of the major regions that are being affected by SARS-CoV, that progressive degeneration, is the orbitofrontal cortex, which is in the part of the frontal cortex, and is actually extremely relevant for cognitive function, for normal cognitive function. So when I look at the brain of my monkeys infected, I see like this, I don't know if you can observe in this image, but I see large blood vessels, and there is a disruptions in the neuronal layers. So we see a lot of astrocytes. It's like the white matter and gray matter is like merging together. And it's that the layers are not well defined as they were before. And this is not only observed in the monkeys. So this is the image from a study from Mount Sinai. This is a brain from a patient. And it shows like how you have this multiple cerebral macrohedges, like they're very similar to, so vascular damage is happening not only in the brain, but is also showing in the heart there. And it shows like multiple spots of brain hemorrhage happening around the brain of this patient. There's also what we see in our monkeys. So the cerebral microhemorrhages can be detected in several different regions. Primary olfactory cortex, secondary olfactory cortex. Again, because they are olfactory cortex doesn't mean that they're only taking care of olfaction. These are highly important regions for memory and high-end cognition for cognitive function. And you can see when you have this microhemorrhages, you have a lot of inflammation, shown here in red as astrocytes. These blood vessels are being disrupted, the astrocytes are being activated, and it's really disrupting the neuronal layers surrounding this blood vessels. Other interesting findings we observe is like myelin degradation. So because as I mentioned, like the virus is really taking advantage of the neuron, the virus really hijacks the neural machinery to like propagate faster in the brain. As a consequence, there is a lot of axonal destruction. So we see a lot of degraded myelin being associated with activated microglia, so neuronal death associated with neuroinflammation as well. We also see neuroinflammation, as I said, so you can see here in this image, again in the orbitofrontal cortex, we see microglia, normal microglia in purple, and astrocytes in green, normal in the left and activated in the right. And it's important to say there are recent studies also showing that microglia, just per se, microglia and astrocyte activation are able to cause cognitive impairment. So not only you have neurons being infected, but also you have neuroinflammation as well. And it's really hard to determine what is causing the other, but they are very correlated and the outcome is very similar. This other image from microglia, and I'm sorry, I have to include microglia because they are my favorite cells in the brain, but it's just showing like how microglia is much more activated and when you have the presence of the virus. And you can see the nucleocapsid protein of the virus being expressed mostly in neurons in this image. And you can see how the microglia, the morphology is much different, and how the microglia are interacting with the neurons is very different as well with the presence of the virus. Additional damages, as I mentioned, blood vessel disruption is one of the major findings we observe as well. So we see this accumulation, and one more time, this animal was not vaccinated, so we know that this spike protein is not coming from any vaccine or anything, it's coming from the virus only. And we see a lot of spike protein and other viral proteins inside the blood vessels, and it's really causing the leaky blood-brain barrier. I think probably a lot of people heard about recently, there was this recent study that was conducted in long COVID patients, showing that long COVID patients have this blood-brain barrier disruption or leaky blood-brain barrier. And this very interesting study, they show exactly the same regions we are observing the viral presence. So the orbital frontal cortex, the piriform cortex, and entorhinal cortex, these are the regions that we have blood vessel disruptions as well. And they are connected to a reduction in the thickness of the brain tissue in these areas as well. Finally, going to the end, I also want to mention that I'm talking a lot about the active viral presence in the brain, but it's not the only cause, or not only way that the COVID can cause neurological dysfunction. There are way more studies, but I just wanted to put this two recent ones showing that indirect damages. So, for example, the one in the left is showing that exposed to the lung-derived cytokines can increase microglia activation in the brain. So when you have impairment in the corticosteroids levels, you can have also an impairment in the pro-inflammatory cytokines circulating in the periphery can really induce microglia activation in the brain, even without the direct presence of the virus in the brain. And this other study in the right is showing that a systemic COVID, SARS-CoV infection can induce dysautonomia by disrupting the vagal nerve signaling as well. So again, pro-inflammatory cytokines can impair brain and cause neuroinflammation, neurodegeneration just by the connections with the brain as well, without necessarily having the viral presence in the brain. So coming to an end, it's like right now the field is very divided and it's all very new. I think, like, of course, viruses in the brain is not new, but I think because COVID and long COVID are so common nowadays that it's challenging the field. We have, like, how can we prepare for the future? How can we prepare for potential new pandemics? Or how can we deal with the damage that it's already causing? Like, how can we deal with a virus that is so neurotropic that infected and affected so many people worldwide? So that, I think, is the major question, and there is a lot of question marks because neuroscientists are really learning and we are lost. And like, how do we deal with all this new information and while so many patients being affected? And finally, like, just to mention, I think a lot of long COVID patients already know that, but neurological manifestations are highly present and highly relevant for long COVID patients. So I think estimated like more than half of patients with long COVID have different types of neurological damages as well. This is a paper recent as well from the beginning of the year showing like different types of most common neurological manifestations and how they can transition from pre-COVID to acute phase of COVID to 6 to 9 to 12 months. And they are very highly variable, but it's very prevalent that they persist, right? The neurological manifestations are not getting clear. I mean, people are not really recovering, and we need to understand why and which parts of the brain are being affected and how can we help patients that have this type of problems and dysfunctions. And finally, I want to end early so I can have time to answer questions, but I want to thank everybody it's all the patients with ME and CFS and long COVID patients, and for watching, for being here, and for helping us scientists to understand what is happening and challenging us every day for being better and try to be better for the patients. For all of us that lost someone to COVID, I also lost someone to COVID. So, and I think so many people lost that it's a it's for all of us. But I also want to give my special thank you to the organizers of this meeting, this incredible meeting. They are all patients as well, and they work so hard. So thank you very much to all of you. And that's it. Thank you.
Okay, thank you very much, Danielle, for your presentation. That was very enlightening. So now we can turn to the question and answer part of the session. And I'll start with the most upvoted question from the community. And that is, do you think that or know if neurological damage because of long COVID is reversible after several years, or will there likely be long-term damage?
Great question. I think it is reversible. I really like to be optimistic, and I think there is reasons to be. Like, the brain is really plastic, and there is so much we think we know that we have no idea what's happening. And sometimes we see like more other diseases, and we see so many big improvements. I do think there is improvement even after many years of damage. Yes, I think it's possible.
Thank you. The next question is, based on your hypothesis, what could be the most likely drug candidates to treat brain fog and cognitive dysfunction?
Yeah, that's a difficult question. Like, in one side, I really see potential for the antiviral like treatments, because I really think it's so hard to clear the virus from neurons and from the brain that I do think is worth trying, especially with some antiviral drugs, as I mentioned, like being able to cross the blood-brain barrier. That's the big challenge. We need to find drugs that cross and then can actually enter the brain. So I think antiviral therapies, it's a good start for treating. And I would like to see some clinical trials for neurological damage for that. Yeah.
Thank you. Our next question. In Germany, we hear about pathologies in Stanford MRI of the head. What pathological changes can you detect in long COVID and ME/CFS in functional Stanford MRI or PET CT? Is this a suitable, objective, and recognized detection method for the neuronal changes?
So there would be which type of CT, like imaging, like scans, right? I do. I mean, especially, of course, the studies that have like the before and after, of course, they're very rare because having an image before. But I think the imaging studies are being proved. It's a challenge because most patients, it's very expensive to do these exams, so a lot of people don't have access to them. But I think they're actually very, very helpful, especially because we cannot apply the techniques I'm doing, like microscopy and these measurements we cannot do without having the brain tissue. So I think biomarkers will be really imaging and detecting markers in the blood and CSF as well, spinal fluid. Yeah. So I think imaging is really a good one. Yeah.
Okay, the next question is TENS stimulation of the vagus nerve device any help in neuroinflammation and with post-viral symptoms in general?
Good question as well. I don't know about viral infections in general. Probably I'm going to check if there are studies, but I know there's several for other diseases that cause neuroinflammation. There are several studies showing that stimulation of the vagal nerve can improve neuroinflammation. There is even a recent study showing like how the vagal nerve stimulation can really improve the connection between the brain and immune system, in the periphery. So I do think vagal stimulation can be a potential candidate for long COVID patients. Yeah, I think it might be a very, very interesting therapy. I don't know if there is any clinical trial going on, but I would love to see one soon as well.
Okay, thank you. Here's another question from the community. Do you think we need to start using language such as cognitive dysfunction or brain injury rather than brain fog? And what difference do you think this might make to how it is perceived and therefore treated?
I 100% think yes. Like, brain fog is such a generic term. Like what exactly means fog? It means forgetting something, or can be like very serious damage, can be, I think brain fog doesn't make justice to brain degeneration. We are seeing neurons dying, we are seeing brain shrinking, we are seeing the ventricles of the brain enlarging. This is what we see in patients with dementia. So, and we are not saying patients with Alzheimer have brain fog. We're saying they have dementia, they have cognitive decline. So I don't like the idea. I think brain fog it's a very way to mask the reality, really. And it makes a lot of people think that the virus are not actually doing directly damage in the brain. A lot of people think that brain fog is all because you have inflammation in the periphery and you have some cytokines reaching the brain. It's not only that, you have the virus inside the neurons, and brain fog doesn't do justice to that. So I do feel like we scientists need to be better, and medical doctors, and we need to use the proper terms as we use for neurodegenerative diseases. Like so we don't tell Parkinson patients they have brain fog. So yes, I absolutely think we need to be using the right terms to bring awareness to the problem. Yeah, I agree.
Okay, here's our next community question. How does the demyelination caused by coronaviruses relate to demyelination in MS? Is there a link between long COVID and MS that you're aware of?
So this is also a very interesting question. Very, I think there's just 1 or 2 studies really looking at that. But there is some, so MS, there is a connection between multiple sclerosis and viral infections, because the virus can do what we call molecular mimicry. I'm not going to the details, but like the virus, it's able to confuse the brain, the cells in the brain, and you have this immune activation in the brain of patients with multiple sclerosis. And there are some studies showing that actually coronavirus can do the same. The virus can trick the immune cells to induce and attack some cells in the brain. So there is some connection. There is some like this overactivation of the immune system in the brain, but it looks like it's similar from the viral infection, like connected to MS, to to what we're observing with coronavirus as well. So, but it's just very new. We don't know a lot yet. It's a very, but some of the similar outcomes. So some of the outcomes are very similar. We do see this immune overactivation of the immune system. Yes, in both diseases, unfortunately. Yeah.
Okay, and here's what I think will have to be our final question. Is there a possibility to measure this neurological damage with a normal MRI, or do you need a special device or process?
I think to an extent, I mean, the thing is that I have the impression that the MRI is going to detect more advanced stages of like more extensive cell death. I think like more like PET scan and other type of imaging exams will be a little more helpful in this case, but I do feel like it can be used. I think it should be used if a neurologist can ask for a patient. I think it's a good start. It's just because we have so few options nowadays. How can we detect damage in the brain? So image is an important one, even if it's one that not always is going to detect a difference for patients, but it's a good start at least to start investigating brain pathology, I think.
Okay, so we've now reached the end of our Q&A session with Dr. Beckman. Again, Danielle Beckman, thank you so much for being here and taking the time to present to us and answer our questions.
Thank you. Thank you very much.